Jessica Li is an associate professor at the Department of Statistics, Department of Human Genetics, and Department of Biomathematics. She's also a faculty member in the Interdepartmental Ph.D. program in Bioinformatics and a member in Jonsson Comprehensive Cancer Center, Gene Regulation Research Program area. Prior to joining UCLA, Professor Jessica obtained her PhD from the Interdepartmental Group in Biostatistics at University of California, Berkeley. She received her BS from Department of Biological Science and Technology at Tsinghua University, China, which is a prestigious university in China. Thank you, Professor, for spending time with us. Let me just first, in this talk, Professor Jessica Li is going to talk about how she uses statistics to quantify the central dogma of biology, which is from DNA to RNA and RNA to protein, right? Protein. We learned about protein an hour ago, and now we're thinking about how we get proteins now. So Professor, please, the floor is all yours. At the end, maybe if we have some time, you guys feel free to ask some questions and we can ask Professor. Thank you, Professor.
Thank you very much for the introduction, Danny. So can you see my screen well?
Yeah, we can see your screen.
Okay, thanks. And so it's my pleasure to introduce my research, one of my research topics to our kids, right? The audience here. So the question is actually in the middle of statistics and biology. So central dogma is known to be a fundamental principle of molecular biology, which I will introduce very soon. So first of all, for our audience, I want to introduce what is statistics. So many people hear this term, but it's often confusing. So I think the term has evolved a lot in the past century, or actually more than a century. Its foundation was a discipline or a branch of mathematics called the probability theory, which was developed in the 16th to 17th century. And it's a branch of mathematics concerned with probability, and it serves as the foundation for statistics. So for example, if we want to study probability theory, we will do a lot of math derivations as shown here in this figure. So it's math. And then later, statistics was developed as a new discipline, stemmed from the probability theory. So we say that it's an application of probability theory, and it studies nature of data and also of the theory of data analysis. So for example, the most famous topic in statistics is linear regression. So let's say that those dots represent our measure values of two variables in those two axes. And the question is to find a relationship between the two variables. This is what we call regression. So in mathematical statistics, which started almost a century ago, we still use a lot of math to solve the problems, and a lot of the solutions were derived by hands. Now, moving into this 21st century, we call this computer age of statistics because we now have the powerful computers to help us do the data analysis. So seeing that this word cloud from Google Images, you will see that data collection, data organization, and some descriptive analysis have become more and more important, not just mathematics. So now the statistics field is considered to be the core of what we call data science, which is about how we analyze data to extract scientific knowledge from data. So I think our field is broadening, and we will need more and more bright young minds to join us in this adventure. Okay, so that's about statistics. And about my own research, my group is titled The Junction of Statistics and Biology. So we deal with biomedical data analysis. So in my field, you see this trendy word, next generation sequencing and precision medicine are becoming more and more popular, and both will generate large data sets that require analysis. So many of the data sets are new, and we cannot directly apply existing methods to them directly. So we have to develop new methods specifically for those new data types. So this is my team. So I think this is an old picture, but we are having new members joining us to do research together. And we took this picture in the launch of our stats department. So one quote I want to give our audience today is that, why do we want to study statistics? The best thing about it is that we get to play in everyone's backyard. That means statistics can be connected to many fields because many fields generate data. And as long as you generate data, we need to analyze data, and then we need statistics. This was a quote from a famous statistician, Jonsson. So I think it still applies nowadays. So back to the title of my talk, the central dogma. What does it say? It is the most fundamental principles of modern biological sciences. So in our cell, the genetic material is called the DNA. But how does this material determine the shape and the function of cells? So it goes through this process called the central dogma. First, the DNA will be transcribed, and this process called transcription, into messenger RNAs. And messenger RNAs will then be translated, this process called translation, into proteins. And of course, besides transcription translation, there will also be degradation processes. So mRNA will be degraded as well as protein. So what we have as the number of proteins of each type in our cells will be a combined result of the central dogma. So we know proteins are the workers that play the roles in our cells, right? So DNA stores the information, but what execute the roles are proteins. So then a fundamental question is, how can we know which steps contribute more to the protein levels we observe in cells? So I want to say that the central dogma was developed more than 50 years ago, but it was until now that we are able to study this process quantitatively, thanks to the development of technologies. So basically, that's what we call high-throughput experiments. So we want to be able to measure how many mRNAs are generated for thousands of genes in our genome. So gene is a unit of our genome. So a gene in the DNA, which is a region in the genome, will become a protein in most cases. And those are the genes we are interested in. So therefore, we want to be able to measure those genes levels at mRNA level, as well as their expression levels at the protein level, so that we can determine, does transcription contribute more, or does translation contribute more to the protein levels we have in cells? So for measuring gene expression at the mRNA level, we have technologies including microarray and next generation sequencing. So microarray was developed in the 1990s, while next generation sequencing was developed around 2005. So both technologies are still being used because they have different advantages in terms of accuracy and costs. So at the protein level, the current technology is called mass spectrometry, which allows us to measure thousands of genes expression levels in protein. And finally, for those degradation steps, we can use pulse labeling to measure how fast mRNAs and proteins degrade. So this project I started working on was from my PhD study in 2011. At that time, there was a Nature paper published. This is a top journal in science, right? So Nature had this paper that measured thousands of genes, mRNA levels, and protein levels, and degradation speeds in mice. So this is the first availability of such massive data on a global scale. So the conclusion from this paper is actually was very shocking, because it says, RNA levels do not well predict protein levels. So in other words, even if we can measure RNA levels, it doesn't mean we know what protein levels could be. This challenged a lot of existing studies in biology. The reason is that our techniques are far more advanced for measuring RNAs than proteins. Therefore, a lot of conclusions were based on RNA levels instead of protein levels. But if this conclusion holds, then those old results based on RNA levels would become problematic. So my collaborator at Berkeley, Dr. Biggin, became interested in this question. We wanted to check whether the analysis in that Nature paper was reproducible and solid. And this was also motivated by a very famous paper published in 2005 that questioned the validity of published research. So it means that even though a lot of findings were published, they should still be checked by third-party researchers again to really verify their validity. So motivated by this, we actually use our statistical expertise to reanalyze the data. And we found two issues that were ignored in the original study. The first is that the original authors treated the data as 100% accurate without considering the uncertainty in those measurements. And that uncertainty is the focus of study in statistics. So by modeling that possibility of uncertainty, especially the mass spectrometry data for measuring protein levels is known to be very noisy. So after accounting for measurement uncertainty, we found the result that it is not certain to conclude that translation dominates over transcription. Instead, we think transcription still plays a very big role. Okay, and we also developed another model based on their data, so using an orthogonal perspective. So this time, we basically used translation rates that are measured by other related studies instead of the translation rates which were inferred in this study, in this original study, because basically they do not have direct measurements, but just some inferred values. So replacing the values by measurements, then we found that the contribution of transcription can be even bigger. So this raises a question about the conclusion about in the original study. At least we think it was too early to confirm, the translation just dominates. It could be case by case. And so therefore, it was also too early to claim that previous results based on RNA level data were not trustworthy. So I think this is a very interesting example to show that we can use statistics to challenge existing findings and offer our new insights. And because of the importance of this topic, our results were included in this book written by Burke is a UCLA professor in this molecular cell biology textbook, which was used by undergraduate students. Okay, so I want to say that the debate is still ongoing. So you will see work supporting the more important role of transcription and work supporting the more important role of translation. So our current understanding is that it's context specific. So we believe that if the biological system or like the cell state is stable, then transcription plays the dominant role. But if the cell is undergoing some rapid changes, such as immune responses, then translation may get into play and play a bigger role, and just to make the cell change faster. So after that, we continue the study in more species, like this is yeast, this is a plant, this is fruit fly, this is mice, this is human. We wanted to see whether we can find some interesting conserved mechanisms about how translation is controlled across those species. Okay, that's my first example. And the second example, which I also find quite interesting, is a topic which is still of crucial importance nowadays, but this was studied by my PhD advisor at Berkeley back in 1975. So it's about the admission gender bias in UC Berkeley about PhD programs. So if we look at the admission rate for men and women at that time, we see that the admission rate for women was much lower, right? So this raised the question about the existence of gender bias. So that's why my advisor at that time, who was a young assistant professor in the stats department, was invited to join a team to investigate this. And interestingly, they found that if they decompose the applicants and admitted applicants by departments, they found that, at least among the 6 departments here, 4 departments gave women a higher admission rate. So this seems to be counterintuitive, right? Why do we have different conclusions by looking at the overall data instead of the by department data? What drove this difference? So if we look at the table carefully, you can see that even though in those departments A, B, D, and F, women did have a higher acceptance rate, but their numbers of applicants were actually very small, right? So the majority of women were applied to department C, and E. So C, D, E, F overall has more people than those 2 departments. And also, if you look at the admission rate or the chance of admission, you can see that the numbers are lower in department C, D, E, and F than A and B. So basically what this suggests is that even though by department, women didn't seem to have a disadvantage, but because more women applied to those more difficult departments whose acceptance rate was low, therefore their overall admission rate was lower. So that was the basic conclusion from this paper. And in statistics, this is a very interesting phenomenon called the Simpson's paradox, which means that the conclusion you draw from the overall data can be different from the conclusion if you stratify your data into categories and examine each category separately. So I think this is something which makes statistics very interesting, one aspect of that. So it shows that analyzing data carefully may give you different conclusions. Yeah, Simpson's paradox. So finally, I want to say that in my research group, our goal is to really find a junction between what we have in theory for statistics as well as the real problems. So we want to be able to apply those nice theory to important problems, in particular in biomedical sciences, so that we can find powerful solutions to those problems. So finally, I want to echo this interesting quote from Turkey again. The best thing about being a statistician is that you get to play in everyone's backyard. So I hope that our audience here can find this topic interesting and possibly consider statistics as your future major. Thank you.
Thank you. Thank you so much, Professor. I love the quote at the end too, and it's so special to see how statistics evolve over time and now into a greater future. Thank you for charting that future for us and explaining how everything is in statistics. What do you guys think? How do you guys think of that talk? Thank you so much.
Thank you.
So we do have some questions in the chat. But before that, I just want to also tell the audience a little bit too, because I'm also in the stats department. If you're interested in more statistics, there's a booth by UCLA Statistics, which I sent a link to. Okay, feel free to check it out. They're gonna give you gifts if you participate in their experiments. I think it's worthwhile to, if you're interested in statistics, to get to know more about how it works. Okay, so we have a question here from Deborah about our COVID vaccine and how it's mRNA.
Right. It is the same mRNA I was talking about. Yeah.
Oh,
it's
it
the
is.
molecule. M stands for messenger. Messenger means that RNA is an intermediate messenger that transfers information from DNA to protein. So yeah. So what we have in the COVID vaccine were we call that exogenous mRNA, which means that these were not produced in our body, but in the labs, right? So they injected those mRNAs into our body. So then these mRNAs will become proteins through the process as a translation. Then those proteins will actually basically those proteins produced by the COVID-19 mRNAs will be something to stimulate our immune response so that our system can generate antibodies again to fight against the COVID-19, the virus. Yeah, SARS-CoV-2 virus. Yeah.
Okay, that's very, thank you. Yeah, that's very, very detailed. Thank you for explaining. Okay, Deborah says, thank you for explaining.
Yeah, so that's why I think so before COVID-19, I was working with my collaborator on how we can predict the translation rate or the speed that each mRNA can be translated into protein. So we're looking for what features does this mRNA have to determine its translation rate. But now with the COVID-19 vaccine development, I think our research become more relevant because we want to have the mRNAs in the vaccine to be more effective so that a smaller amount of mRNA can give us a large amount of that beneficial proteins. Then that can help us fight the virus.
Wow. I think it's so cool that, Professor, it's like you said, like how statistics connects with so many other aspects.
Yeah.
Would you say that how did you first get into the junction between biology and statistics?
Yeah, I think, I think my journey was quite interesting. I was fascinated by science when I was very little, and my parents were both majored in mathematics, and they were high school mathematics teachers when I was young. And so I think I like mathematics as well as science. So when I was thinking about my major for my college, and my parents told me probably mathematics is already a very well developed discipline, but biology is still much underdeveloped. So a lot of questions remain open. So I feel like, I wanted to do, if I want to do something related to science, biology is a very rich, a very rich mind, which hasn't been really explored. So that's why I chose to study biology in my undergrad major. But quickly, I realized that biology was still primarily experimental discipline, that people focused a lot on data generation. But at that time, microarray were already fully developed. So I can see that large data sets can be generated. So I realized that, since I like math, I think math skills would be needed by biologists in some day, just like physics and chemistry, which were first started as experimental observational studies, but quickly they need math to formalize their hypothesis and theory. So that's the way I see biology to go. So that's why I decided that I would need more quantitative skills to do the research I want. So that's why I chose to study statistics for my PhD. And then I came to UCLA, and my research is now at the intersection.
That's amazing. Yeah, it's like a process, like a natural process from science, as you get interested and as you find more need, you grab onto more knowledge, which interconnects with each other. Yeah,
I think the passion is the biggest drive.
Yeah, thank you so much for the talk, Professor Li, and for your passion on the topic.
My pleasure.
Really appreciate it. Yes, so let's see, do we have any concerns or any thoughts, comments for our talk? We still have a couple more minutes. We just want to give everyone a chance to also voice your opinion before we move on, right? Okay, I think we're good. Okay, well, thank you, Professor, so much.
Thank you,
guys. please just, if you really appreciate the talk, let's type thank you to Professor in the chat, okay? And we appreciate her for spending her time today. Thank you, guys. Thank you, Professor.
Thank you.
Have a good rest of your day.
Thank you.
Thank you. Ryan said thanks. Carla said thanks. So Shongham said thank you. Calvin, thank you. Warren, thank you. Awesome. Thank you, guys.
Thank you.
Okay, so we're all